First of all, thank you very much to the organizers of this workshop for inviting me to present here. I have to say, listening to all of the stories on the human impact of wildfires is sobering as a scientist. Let's see if I can get this here. Okay. So I'm clearly the outlier of this session, is probably also the workshop, in the sense that I'm going to be talking about the effect of wildfire smoke on an animal population. And so the picture on the slide should be your first clue. I'm going to be talking about the outcome of some studies that have taken place over the course of the past 10 years, believe it or not, looking at the effect of naturally occurring or ambient wildfire smoke on a cohort of non-human primates, specifically rhesus macaque monkeys, that live outdoors at the California National Primate Research Center. Okay, so I'll start off with the impetus for this study. So when I first conceived of this study, to be quite honest, I wasn't considering or focusing on wildfire smoke per se. The impetus for the study was really to ask a very broad question about exposures to air pollutants and the type of exposures that these animals were experiencing. And so the exposure event that took place and is the focus of our study is one of the wildfire events that Colleen mentioned, which, by the way, this is a perfect introduction. Your introduction was perfect for what I'm going to talk about. The 2008 fires, consisting of the Humboldt, Trinity County, Northern California fires, which brought in a significant amount of smoke into the Sacramento Valley, and specifically in Yolo County, where our primate center is located. So when this event took place, I have to tell you, I was sitting in my office, I looked out the window, and it looked like fog in the middle of summer. And the light bulb went off in my head, and I'll mention why in a moment. But the first question was to simply ask whether this type of acute exposure event had a health outcome in our colony. Now, in comparison to what we know from the literature, we know that this isn't a surprise. We know from the human literature, as well as in experimental animal studies, that chronic exposures to high levels of pollutants are bad. They have detrimental health outcomes. We know that acute exposures have these short-term health effects, as Colleen has mentioned, but we really don't know whether these acute events, like what's shown here in the slide or in the image of the Santa Rosa fires, we don't know whether these acute events have long-term health outcomes. So we're going to use, or what I propose to do to the California Air Resources Board, is to use this event to study the long-term effects of an acute exposure event in a susceptible population, and specifically very young animals. So we asked the question of whether the short-term acute exposure events, as that observed from a wildfire smoke event or a wildfire event, could result in long-term health outcomes. So this question was addressed by two overlapping events. So as I mentioned, the first event was the Humboldt, Trinity County, California wildfires in the summer of 2008. This overlapped with another unique event at our California National Primate Research Center. So the spring and summer are what we refer to at the center as baby season. So rhesus macaque monkeys are unique in that they are seasonal breeders. They breed in the fall and they give birth in the spring. So in the springtime, we get anywhere from 500 to 700 infant monkeys outdoors. And during this time, we're being exposed to the wildfire smoke. So just again, as background and as a reminder of what took place in 2008 from the Humboldt, Trinity County wildfires. So this was due to a lightning strike that took place towards the end of June, and the wildfires continued through the early part of July, and it brought a significant amount of smoke into our area. So I don't know if you guys can see the red dot. The red dot is actually our little town of Davis, California, where the University of California, Davis is located. So fortuitously for us as researchers, the California Air Resources Board has a number of monitoring stations throughout the state of California. One of them is located on the UC Davis campus. And in fact, the monitoring station for which the data that contributed to this graph is located actually about 2.5 miles from the California National Primate Research Center. So we were actually able to get some real hard data in terms of exposure levels relative to our center. So as you can see here, I don't know if this pointer works, but on the graph here, clearly there are 2 peaks of PM 2.5. This is a 24-hour average throughout from June 1, which is at the to your left-hand side, and July, at the end of July on your right-hand side. So there are 2 very significant peaks of PM 2.5 within the primate center area in comparison to the dark line, which is on the bottom. That represents data collected in 2009. So clearly the animals that were born within in the year 2008 were exposed to a significant amount of PM 2.5. So I wanted to give you a visual of the footprint of the center, primarily because the public perception of primate centers is that our animals live in isolation, and that couldn't be further from the truth. So this footprint shows you a map of the California National Primate Center. We're about 100 acres sitting outside of, or on the outskirts of UC Davis. We have at any point in time about 4,500 rhesus macaque monkeys on site. It's a pretty impressive facility, and about half of them live outdoors on a 24/7 basis. So we are a 365-day operation. So, as I mentioned, our animals, many of our animals live outdoors. Over on your left, you can see a visual of our half-acre field cages. So our animals live in large social groups. They breed out there, and they play out there, just like children do on play structures. So the first part of this talk will cover the first study that we conducted and with the support of the California National, not the California Center, the California Air Resources Board, and was published a couple of years ago. And then if I have time, I'll be talking about the completion of a second study, which we've not yet published. So in this first study, as I mentioned, we wanted to study the health outcome of wildfire smoke coming into our area on our very young animals. And so instead of studying the animals immediately after birth, we actually studied the animals when they were adolescents, so they were 3 years of age. So we really are looking at the long-term health outcome of an early life event. And so we studied the animals that were born in 2008 and exposed to this event. And then as controls, fortunately, the year after, in 2009, we had a relatively clear air quality period during the summer. So we were able to use the animals born in 2009 as a control. And then I should also mention that we looked at both males and females in this cohort. So, so what did we do? So in our proposal to the California Air Resources Board, we used the approach that the evaluations that we conducted on these animals would be non-invasive. And this was really important for us because we wanted to be able to have our data or allow our data to potentially be extrapolated into the human population. So we needed to use approaches that could be conducted in the human population. So we did 2 things, and we proposed to evaluate 2 systems. So we proposed to evaluate both the immune system as well as the respiratory system. So for the immune system, we collect peripheral blood, and then we took it back into the laboratory, and then we challenged enrich the blood in the laboratory with components of bacterial cell walls. And this was done to mimic a bacterial infection. So we didn't want to infect the animals directly. We just wanted to see whether their blood could respond to an infection. In addition, these animals underwent pulmonary mechanics testing, including methacholine challenge, which is a measure that helps us to assess for development of asthma. So what did we find? So what these two graphs show you are data collected from our blood studies, and I realize that this is a, this is very targeted for immunologists, but I think you can see a difference between the white bars, which represent the animals that were born in 2009 are controls, and the black bars, which represent blood samples from animals that were born in 2008. And so our outcome or our readout measures were proteins that are elicited in the immune system in response to any type of bacterial challenge. So these proteins are IL-8 and IL-6. So the take-home message here is that the immune system, that the animals that were exposed to the wildfire smoke very early on in life, 3 years later, had an immune system that was suppressed in its ability to respond to a bacterial infection. In addition, with our lung function testing, you can see on your left-hand side, I'll try to keep this very simple. The two bars that you see on the left-hand side represent overall lung volume. So the overall lung volume of animals that were exposed to wildfire smoke very early on in life was reduced, significantly reduced. In addition, over on your right, when we measured for lung compliance, which is a measure of stretch or overall lung stretch in these animals, we found that the animals exposed to wildfire smoke had reduced compliance. So what does that mean? That means, or that could be translated as a very stiff lung or significantly stiff lung. Now, what we did not find is evidence of asthma, so no airways reactivity in these animals, again, at 3 years of age. So moving on, we were able to follow up on these animals when they reached adulthood. So this is all the way up to 8 years of age. And in this case, we specifically focused on females. So why did we focus on females? So if you, I don't know if you caught it in the previous graph, it was the females that elicited the most significant response with regards to lung function. And also, we were interested in studying the offspring of these animals. I'm not going to have time to talk about the offspring, which are really interesting, but suffice it to say, we decided that females would be the most informative population for this adult study. So again, we used our 2008 cohort, compared it with our 2009 cohort as controls. And so looking at the immune system in a similar way, collecting blood and looking at lung function, we find that, in fact, the immune system remains dysregulated 8 years after the exposure, 8 years. And these are animals that are living outdoors, again, under normal conditions. So to study the lung function in these animals, we took a slightly different approach, and this is, I think this is pretty cool because it really takes advantage of some very cool technology. We have a PET CT scan on site, so this is just what humans can undergo for diagnostics. So our animals underwent CT scans, and we collected the images, and then we had the images analyzed by a company called Fluida. So they partnered with us to do image analysis of the CTs collected here, and they were able to use machine-based learning to do some very sophisticated analyses of lung function as well as airways remodeling. So what did we find? So at the very top, what you see, well, hopefully you can see on the right-hand side, the wildfire group animals had a significantly reduced lung volume, consistent with what we observed in juveniles. And you can see the images, which is a compilation of all of the animals in the study on the right-hand side. On the bottom, we see also a reduced inspiratory capacity, again, a measure of reduced lung function in these animals. And last but not least, I'm not going to have time to go through all of the data we collected, but suffices to say we observed significant differences for many parameters of lung function in our animals at 8 years of age. We also observed changes in overall structure. At the very top, what we're measuring is airway radius, and surprisingly, the animals that were exposed to wildfire smoke early on in life had enlarged airways as well as increased vascular density. And you can see that on the very bottom. You can see the branches, the branch points, and the different colors which represent enhanced blood vessel growth within the airways of these wildfire-exposed animals. So in summary, I know I threw a lot of data at you, and I apologize, but I wanted to hopefully emphasize to you that, in fact, something that takes place very early on in life can have long-lasting and profound effects on the respiratory system as well as the immune system. So in infancy as well, or in adolescence as well as in adulthood, we see what we believe is very strong evidence for immune dysregulation, and we clearly see structural changes to the lung and functional changes to the lung. And so what I hope I've convinced you is that clearly there's an effect of early life exposure, and that what we would propose is that our animals could potentially be used as surrogates for health outcomes in susceptible populations following a wildfire event. And I'll end with acknowledging the contributions of the California Air Resources Board. Deborah Drexler, who retired a couple of years ago, was really a champion for this project. So I want to acknowledge her, and her role has been taken over by Lori Miyasato, who's also here from the California Air Resources Board. Thank you very much.